This recording will start Chapter 3, discussing cell structure and function. Let's take a look at our outline for this chapter. We'll start by listing the main points of the cell theory. We'll then describe the functions of the plasma membrane and the structures that enable it to perform those functions. We'll be describing the process of cellular diffusion, osmosis, and filtration, and explain their physiological roles. We'll be describing carrier-mediated and vesicular transport mechanisms and explain their functional roles in cells. In section 3-5, we'll describe the organelles of a typical cell and indicate their specific functions, including that of the cell nucleus. We then get into summarizing some important processes, like the process of protein synthesis in section 3-7. And then in 3-8, we give you a warped tour of the stages of the cell life cycle, including mitosis, interphase, and cytokinesis. And then we will briefly discuss cell differentiation, section 3-10, and explain its importance. So just as atoms are built out of protons, neutrons, and electrons, complex organisms such as yourself are built up of cells. And over the decades of study, scientists have developed the cell theory, which states that cells are the building blocks of plants and animals, that cells are the smallest functioning unit of life, cells are produced through division of pre-existing cells, and that each cell is responsible for maintaining homeostasis. Therefore, if you want to understand the human body as a whole, you must become familiar with the nature of a cell, which is why we then continue our discussion of general A&P by looking so closely at the cell. And so the study of the structure and function of the cells, so this chapter is cytology. This whole chapter is about cytology, the study of the structure and function of a cell. And now a branch of science which combines knowledge about cells from microscopic study, chemistry, and physics. So cytology really does take a look at the typical cell and its components. So the cell pictured in Spotlight figure 3-2 doesn't really exist. It's an artist's design that kind of shows many of the common features of a cell, but there's no one cell that actually looks exactly like that one pictured in that figure. In reality, cells in the body are actually very diverse, and each type is built for a very specific function, like these pictured on this slide. So they have many different shapes and a variety of special functions. The cell types shown here have the dimensions they would have if they were magnified about 500 times, and you're looking at them with one of the light microscopes like the ones we'll be using in lab. And so you can see there's certainly a difference in the structure of, say, a sperm cell compared to that of a neuron or one of the cells that make up your brain. And just based on their structures, you can see why they have different functions, right? And that of a fat cell or bone cell or blood cells. All of these cells have different structures because they have different functions in the body. So we're at a checkpoint here, and I'm wondering if you can answer this question. What are the 4 basic components of the cell theory that I just told you about? Can you pause the lecture recording and see if you can recall those? Now the next section, 3-2, discusses the plasma or the cell membrane of the cell. And this is an important structure because it separates the cell contents, known as the cytoplasm, from the typically watery environment of the external world. It contains lipids, proteins, and carbohydrates. So the 4 key functions of the plasma membrane or the cell membrane, those 2 terms can be used interchangeably, is that provides the cell with that physical isolation from the extracellular world or the extracellular fluid. It regulates the exchange with the external environment, and it allows for sensitivity to the environment. That's one of the ways that cell-to-cell monitors what's happening in your environment. and it provides for structural support of the cell. So those are some very important functions of that cell membrane. Let's take a look at an artist's depiction of the plasma membrane. Note the membrane lipids, membrane proteins, and membrane carbohydrates that are present. So lipids are the major components here. The phospholipid with its hydrophobic head, its hydrophobic tails here and its hydrophilic heads. Notice how the hydrophobic tails are facing each other. And this is going to create a phospholipid bilayer. So here's a layer of phospholipids, here's a layer of phospholipids. So we call it a phospholipid bilayer, and it's labeled right here for you. And this is a very fluid, dynamic, flexible structure. It's not at all rigid. And actually, that's one of the reasons we have cholesterol molecules mixed throughout in here. Here's a cholesterol molecule. These are going to add some rigidity, these little like orangey colored squiggles. Those are to represent cholesterol molecules, and that will add some stability and rigidity to that lipid structure. So lipid-soluble compounds and gases can easily cross the lipid part of the membrane. And there's also many different kinds of proteins associated with the membrane that are pictured here. Some are going to float around on the membrane surface. Think of like an inner tube floating around the surface of a pool. Others are going to span the membrane, like this one is spanning the membrane. Here's one that spans the membrane. And these are called transmembrane proteins. And these can be anchored in place. So instead of just floating around, they can be anchored in place. So this one is anchored. This one probably floats around since it's not anchored here anywhere. Carbohydrates often combine with proteins and lipids to form glycoproteins and glycolipids. So we have a carbohydrate chain here. This looks like some carbohydrate chains here combined with these proteins. And so what these glycoproteins and glycolipids can do, they can function as lubricants and adhesive receptors, even identifiers for cell-to-cell communication. So when we discuss the membrane, we're going to be talking about its permeability. So let me define permeability for you. Permeability is that property that determines what substances can cross the membrane. So for example, we could say a membrane is impermeable, meaning that nothing can cross that membrane. Or perhaps the membrane's freely permeable, where anything can cross. So those are two different varieties of permeability. Our membranes are what we call selectively permeable, meaning that some things can cross the membrane while other things cannot. So it's selective about what crosses the membrane and which doesn't. And these movement processes can be either passive or active. Now, I'm going to start with passive movement processes first. Passive movement processes use kinetic energy and typically are going to proceed to equilibrium. Whereas active processes, on the other hand, typically require cellular energy to be spent. Typically, ATP is going to be burned in an active movement process. And there's two types of passive processes, which are diffusion and filtration and osmosis. So osmosis is actually a type of diffusion. So let's take a look at those different processes. And we'll look at those in a moment. But first we have this figure that shows you the idea that a path the substance takes in crossing the plasma membrane depends on that substance's size and whether or not it's soluble in lipids. Now remember, like dissolves like. Okay, and I told you that lipids are nonpolar and they're hydrophobic. So other nonpolar hydrophobic substances can get along with the lipids and they'll just diffuse across that lipid membrane. But that is one of the reasons that we have these channel proteins for those things that are polar and will not dissolve in a lipid membrane. Like this steroid, perhaps, which is pictured here, is just diffusing across the membrane. All right, so I promised to talk about the different movement varieties. We'll start with diffusion. Molecules that move from an area of high concentration to an area of low concentration are said to be diffusing. And since this movement occurs down a concentration gradient, there's no need for energy to be associated with diffusion. It's energy free. Diffusion is diffusion. It's diffusion. It can occur in a general area, which is pictured here in this beaker, or it can also occur across the plasma membrane. So lipid-soluble molecules can pass across the lipid portion, the phospholipid bilayer, of a membrane easily. And the example I gave you in the last picture were steroids or hormones, for example. But the non-lipid-soluble molecules like oxygen, carbon dioxide, or ions, those have to diffuse through the channel proteins. And water has a special channel protein called an aquaporin that it diffuses across the membrane freely. So speaking of diffusion of water, that is called osmosis. So water creates this special case of diffusion called osmosis. And in our body, water is probably the most common solvent in which other substances called solutes are dissolved. So there's three characteristics of osmosis. The first is that diffusion of water molecules across a selectively permeable membrane. So it's diffusion of water. The second is that osmosis occurs across the selectively permeable membrane that's freely permeable to water, but not freely permeable to solutes. And the last characteristic of osmosis is that water flows from low solute concentration to high solute concentration. And so this is called osmotic pressure, the force of water of one solution moving into another solution towards the higher concentration of solutes until equilibrium is reached, at least. And so oftentimes when we're talking about solutes, we're referring to salt. And so as a rule of thumb, by osmosis, wherever the salt is, water will follow. And that's what's being pictured in this next upcoming sequence of figures, is that we have these two solutions that are separated by a membrane, and the water molecules are pictured in blue here, and the solute molecules are pictured as little purple balls. Now, clearly, there is more water on this side than this side. And so to try and reach an equilibrium, and there's more solute on this side too, so what water will do is it wants to follow. Wherever the salt is, water will follow it. And so the level of this volume will increase until we've reached an equilibrium, meaning that the amount of solute in water is the same on both sides. So you can see the concentration has really equaled out here. And so water is moving by osmosis across that membrane until a equilibrium has been reached. And so you might be asking yourself, why does this matter? Well, because solute concentrations in the movement of water have an effect on the shape of your cell membrane. And we describe this effect as in terms of tonicity. So an isotonic solution is one that has the same concentration as that of your intercellular fluid. So there's no net movement of water across the membrane, meaning that water is still moving, but it's equal. It goes in at the same rate that it's coming back out. And so the cell membrane remains intact. Whereas in a hypotonic solution, if you drop a cell into a hypotonic solution, this is going to be a solution whose concentration of the solute is lower than that on the inside of the cell. And so water is going to move into the cell. Okay, because right now the inside of the cell has more solutes, more salt, than its hypotonic solution around it. And so water is going to follow the salt into the cell. It's going to cause the cell to swell, maybe even rupture because it's so full. So we can see this in red blood cells. It's called hemolysis. And this is one of the reasons that even when someone is dehydrated, you do not run pure water into their IV, right? What do you run into their IV? Have you ever looked at what it says on that bag? It says saline solution. Do you ever wonder why you give salty water to someone who's already dehydrated? Well, that saline solution is isotonic with the amount of salt and sugars and other solutes inside of the red blood cells. If you were to run straight water into someone's vein, their red blood cells would lyse. They would experience hemolysis of their red blood cells. So now the last tonicity solution that I need to tell you about then are hypertonic solutions. So hypertonic solutions are those which the concentration of the solutes is greater than what's inside of the cell. And so water will follow the salt out of the cell. Water's going to leave the cell and go out to where the greater concentration of solutes are. And so the cell will shrink or crenulate. So we have a figure of what this looks like to your red blood cells. So here's an isotonic solution where the concentration of salts and water and sugars and solutes are the same on the inside of the cell as they are on the outside. And so water's moving out and in at approximately the same rate. There's no net movement. Here the net movement is in. So this is clearly a hypotonic solution where water is going into the cell because there's more solutes. See these little purple dots? There's more solutes on the inside of the cell than there are on the outside. Okay, so this would be like dropping a red blood cell into a completely distilled water. Okay, so this is an actual scanning electron micrograph picture of what a red blood cell looks like when it's been dropped into a hypotonic solution. And then last, in a hypertonic solution, so if you were to drop that red blood cell into, like, say, a 10% glucose solution, you would get this crenulation of the cell where it's going to shrink because all the water is going to be leaving the cell and going out towards all of the solute on the outside. And so your red blood cells actually shrivel at this point. They lose their volume. So another type of passive process to discuss, other than diffusion and osmosis, is filtration. Okay? And so filtration occurs when the hydrostatic pressure forces water across a membrane. So if solute molecules are small enough to fit through the membrane pores, they'll be carried along with the water. And we're going to discuss filtration in much more detail in the urinary system chapter when we talk about the processes involved in the kidneys filtering your blood. And so we've reached a checkpoint box, the last one of this particular recording, and I'm wondering, do you know what's meant by selectively permeable when referring to a plasma membrane? So go ahead and see if you can answer that question or review as necessary, and then you can pick up in section 3-4 in the next recording.